All right, everyone, can you hear me? And can you see my screen? All right, so let's get going. So I wanted to say a little bit about this protein calmodulin because it appears a few times in my lectures. And normally, as I said, I talk about this quite extensively in Biochemistry 431, but other people have been teaching that lately, so you might not have heard about this protein. So calmodulin is probably the most important calcium binding regulatory protein in our cells. It is found in all eukaryotic cells. So all eukaryotic cells make a large effort to pump the calcium to the outside. And only during stimulation of the cell will they actually allow the calcium to flow back into the cell. And then that calcium signal needs to be translated into various responses. And in order to do that, there are these calcium binding proteins. And calmodulin is very important amongst them because it has at least 300 different target proteins that we know of. Calmodulin is a relatively small protein. So if there's no calcium in the cell, this is what it sort of looks like. And then when calcium flows into the cell, you see that it combines 1, 2, 3, 4 calcium ions. And if you look at the structure, you can see that in all cases, the structure is a helix, a loop that wraps around the calcium, and another helix that comes out. And this loop, helix motif is called the EF hand, after another protein in which this was originally discovered in the crystal structure, where this was the E helix, this is the calcium binding loop, and this is the F helix coming out. So you can remember it quite easily with this diagram, what the EF hand refers to. So normally these two occur together. So you have two EF hands, there's a helix coming in, a loop here, and then a helix coming out, a helix, loop, helix. So you have two of these EF hand motifs coming together, and they like to form and snuggle up against each other. And in doing so, they like to form a tiny small beta sheet just between these two calcium binding loops. So these two calciums actually know about each other because there's hydrogen bonds here between these calcium binding loops. And so this is through a small, very tiny beta sheet that forms here. And the same in this other half of calmodulin, you have two of these loops and they also get together. So in total, calmodulin has a lot of EF hands. Every lobe has 4 EF hands, 2 EF hands, so there's 4 of them in total. So there's 8 helices and 4 calcium binding loops. Now after this is bound calcium, what will happen is it exposes some hydrophobic surfaces. And these hydrophobic surfaces allow it to interact with various target proteins. And they have calmodulin binding domains where they bind. And you see they actually induce an alpha helical structure into regions of the target binding protein. And these green things are what we call anchor residues. So there's an anchor residue here sitting in the C lobe of the protein, and there's a somewhat smaller, but also hydrophobic anchor residue sitting in the N lobe of the protein. So there's a tremendous literature on calcium calmodulin, and many complexes have been solved with very different sort of interactions with different target proteins. MLCK is for one of the early ones, myosin light chain kinase. Peptide is the P here. This is only a peptide of the whole protein that was studied in this particular case. Now, calmodulin will come back a few times, so just remember that it's a calcium binding protein. And that it has this unique helix loop helix structure. If you look at this, there's a mostly helical structure, but there's this tiny, tiny beta sheet that forms between the two loops. All right, now the other thing that we're going to talk about is that according to the crystal structure, this is a continuous exposed helix between the two lobes. But according to NMR, this is not a continuous helix. And there's actually a flexible region here in the middle of the protein and I can show you the evidence for that later. So in solution, there's actually the two lobes of the protein are not rigidly held and fixed, but they're actually flexible, and they can take up whatever position they want with respect to each other. And that is one of the secrets why calmodulin is actually so good at activating so many different proteins, because it can take up all these different orientations. Now, we discussed that you can calculate the structure. So this was the structure calculation that we discussed at the end of Monday's lecture. This is for one lobe, so one of the two portions with two EF hands in it for a plant calmodulin. And you remember that we use these NOEs, these hydrogen-hydrogen distances between 2 and 5.5 angstrom to actually calculate that structure. And we started in this particular case out with a completely extended structure just to be unbiased, right? We didn't want to start with a structure that was already close to what it was, but we wanted to be totally unbiased. So let's make it totally extended. And then we put all these NOEs on there. And so you can see some of them are really well stretched beyond that 5.5 angstrom limit. And then we gave it to the computer, who then combined it with some molecular dynamics force fields. And then it calculates the structure. And all the white lines in here are now well within that 5.5 to 2 range. And so this is how you calculate the structure. And of course, the more of these NOE distances you have, the better defined your structure is going to be. Now, this is the structure that came out. This is the structure of one lobe of this plant calmodulin. And as we also discussed, we calculate the structure usually several hundred times. And then we take the lowest 20 energy or the lowest 10 energy structures. That differs a bit from what people's preference is. And you can see here in blue are these alpha helices. And this is the way you're used to seeing these structures. So this is the one of the structure, usually the lowest energy structure. And you can see helix, loop, helix, helix, loop, helix. And there's a tiny beta sheet between the two calcium binding loops is indicated up here. And that's reflected. This is the same orientation of the 10 NMR structures. So you see that beta sheet, that tiny beta sheet of 2 strands only between the two calcium binding loops is easily found. And you see the typical helix loop helix structure and helix loop helix structure. And this, of course, is the linker between the one group of EF hands and the other, one EF hand and the other EF hand in this domain. So this is a fairly typical sort of structure calculation. You see that you get good overlay, so that means the root mean the root mean square deviation across the backbone in this case was calculated as 0.49 angstrom. And usually when you get into that range, your structure is good enough that you can publish it. Now, if you have more data, of course, you try to refine it a little bit better. We have published structures which were defined all the way down to 0.2 angstrom root mean square deviations. But for some bigger proteins where you don't get great data, sometimes you have to be satisfied with 0.7 RMSD angstrom. So it's important to remember always that NMR then calculates many possible structures, because every time you calculate it, because it's a molecular dynamics calculation in which certain things are randomly assigned, the structure does not always come out exactly the same. Now, of course, the structures are widely different, then you don't get good agreement in the RMSD. So the better the RMSD fits together, the better you are doing. Now, as I already said, if you want to calculate the tertiary structure of a protein, because that's what we just did, I only showed you the secondary structure, the helices and the sheets. But of course, we also position the side chains in here. So that's, of course, the tertiary structure of a protein, as it's called. So we also have the side chains in there, and they're also positioned simply by NOEs. and proton-proton distance information between the various side chains. And normally we say that if we have about 15 NOEs per amino acid available, then you usually end up with a pretty high quality structure. Now, apart from NOEs, there's other information that we can also include in the structure calculation. And we've already talked about the fact that in the chemical shifts that we see for the alpha hydrogen along the backbone of the protein, we can see if it is shifts in one way, they're usually involved in a beta sheet. And then if it's shifted in the other direction, it's involved in an alpha helix. So that sort of information, when I say alpha helix or when I say beta sheet, I actually mean that I have different phi psi angles along the backbone of the protein. You've heard about the Ramachandran diagram that specifies certain angles for an alpha helix and for a beta sheet. We can take the information that we have from chemical shifts and also from another phenomenon called spin-spin coupling, which I will discuss later. And actually, based on that, we can give some direction to the backbone and say, okay, you are folding in an alpha helical structure. You are folding in a with a phi psi angle that's more consistent with a beta strand or a beta sheet. And also, in this whole process, we can actually sometimes and often figure out where the hydrogen bonds are, right? The hydrogen bonds between the backbone NH and the carbonyl, those are the hydrogen bonds that define the alpha helices and the beta sheets. And if we know where they are, we can put that hydrogen bond in also as a restraint in our calculation. And so these are the three most important ones: the nuclear Overhauser enhancement effect. So that's proton-proton distances, something about bond angles along the backbone. There's software for that. And then there's hydrogen bonding that you may have found during your exploration of the spectrum. Nowadays, there's another parameter called residual dipolar couplings. I will not discuss those. It's a rather complicated experiment, but it's very useful to refine the structure further. And it's particularly useful for long-range orientation. So if you think about the two lobes of calmodulin, they're quite far apart from each other. And of course, they're beyond the distance that you can measure with NOEs. They're way beyond that 5.5 angstrom distance. So they're not well positioned with respect to each other because you don't measure anything that has that distance. And the nice thing about these residual dipolar couplings is that they give long-range information. So they allow you to really position two lobes that are far apart from each other in space quite well. All right, so that's why that's useful. Now, this is, of course, something that you don't do on your pocket calculator anymore, you know, and so there are computer programs that do all this. And so you won't be surprised to hear that the computer program uses NMR parameters, things like NOEs, like the proton-proton distances. It uses spin coupling data, and it translates those in certain ranges. It says, okay, this bond has to be between this and this. This distance has to be between 2 and 5.5 angstrom, right? So it takes these NMR data and it translates them into a series of constraints or restraints, as we call them often. And then, of course, we have standard bond lengths and bond angles that we know from all the structures that are available in the protein database. And from that, we can start calculating the structure. And in order to do that, there's a certain molecular mechanics force field that's involved in that. And so that gives you then an initial starting structure. And then you look at this structure and say, does it look good? And usually the first time you try it, the first answer is no. You may have misassigned a few peaks, a few things were not correct. And so then you say, well, structure is not really good. There's a couple of errors in there, so we have to go back. You have to fix those errors, and then you can start the process all over again. And then you can say, well, you know, you do this, you go through this process a few times, so it's a bit iterative, and then at some point you get a structure. You say, you know, I'm actually quite pleased. I think this structure reasonably fits with my constraints. And then you have to decide if it's refined enough to deposit it in the PDB and publish the paper. But you can also go back and say, nah, I don't really like it. Maybe we can try a few other constraints. Maybe we can try another method of energy minimization before you decide that you really agree with the structure. And then it's refined enough. You've calculated 200 structures. You take the 20 lowest energy ones, and those you can deposit in the PDB, and then you're done. So there's a little bit of an iterative process, and it takes some time, but these are all standard software packages that guide you through the process if you work on this. And here's an illustration of what the number of restraints actually does to the NOE calculation. So if you only have 7 restraints per amino acid, then you can see this is only the backbone structure of this protein. It looks pretty much like spaghetti. You can see that there's a little bit of that, there's some fold to it. You can probably see that there's an alpha helix there, but it's not overlapping very well, particularly in these regions. And that is simply because we just don't have enough information to really tie things down properly. And you can see in these cases, if we go down here, I should put my laser pointer on. If we go down here, you can see now the backbone is starting to overlap better and better as we add more and more restraints, right? You see in this particular case, there's almost no deviation between the backbone of the 20 different structures that were calculated. And you can also do this for the side chains. They're shown on this side of the diagram. You see here, for example, this is clearly positioned as a tyrosine side chain, which is very well defined in terms of where it is and how it is fixed into the structure as well. So if you have about 15 or 16 restraints, this is sort of quality that you can obtain. This is a bit of a guideline, because if you think about a glycine only has 1 hydrogen on the side chain, and it wouldn't have anywhere near to 15 interactions because it's just not big enough to interact with a lot of things. If you think about something like a tryptophan, which is the biggest side chain of all the amino acids, it usually has well over 20 interactions with all kinds of other amino acids that you can define. So this is a bit of a general guideline, and then the structures that come out are equivalent to what the crystallographers would call a 2.5 angstrom X-ray structure, right? So a pretty well-defined structure. So this is the summary of what we've been talking about, right? And when you see an NMR structure presented, right, it looks different usually than a crystal structure. Although if they only present the lowest energy structure of the 20 that are deposited in the protein database, then it looks like a crystal structure. But if you look at the 20 and you project them all on top of each other, such as done in this diagram here for this particular protein, and there's different side chains indicated in different colors. Usually red is negatively charged, usually blue is positively charged amino acids. And then you have here is some black. Those are probably aromatic ones, and then the yellow ones are probably more hydrophobic aliphatic ones. And you see this is a pretty well-defined structure, but something that we've also talked about already. Usually the ends of the protein, the N-terminal and the C-terminal ends, are not necessarily extremely well-defined because they tend to be a bit floppy, and so that's often the case and discovered by looking and comparing these 20 conformers. Now, when people did the first structure of calmodulin, they started to overlay their structures as well. And you can see this is the structure of calmodulin. And what they did is they overlaid the C lobe of the protein. And you see the C lobe of the protein overlays very, very well, right? But if you look, and this is the helix that connects into the N lobe. But if you look at the N lobe, it looks like a total ball of yarn, right? It's a complete mess, right? A ball of yarn that the kittens have played with. Now, if you do the opposite experiment, however, you overlay the N-terminus, you see it's actually very, very well defined. And now the C looks like spaghetti, so to say. Now, the reason for that is very simple. The lobes, this lobe of calmodulin is very well defined. This lobe of calmodulin is very well defined. But this region in between here is not fixed and not defined. So if you overlay all this N-terminus can take up whatever orientation it wants. If you overlay the N-terminus, of course, when this is flexible here in the middle, the C-terminus can take up whatever position it wants. So it is what I said earlier. You have two well-defined domains or lobes of the protein indicated by my two fists, but how they are oriented with respect to each other in solution is not fixed, and so it can go wherever it wants. And so this is another example of how it's important to work in solution, because this is the salient feature of this protein, that it's flexible in this region, which did not come out in the crystal structure because they had to crystallize the protein and used weird organic solvents to get the protein to crystallize. And in doing so, they artificially stabilized the helix through this particular region. But it's really not a helical region in solution. All right, so far I've only talked about proteins that we've studied with proton-proton NMR. And those, when you purify protein, it doesn't matter from a bacterium or from humans or from whatever bovine material you may have available. Those protons are, of course, in there because remember, 1H is present at 99.9% or something like that. So, yeah, if you want to do proteins, structures for proteins that have more than, say, 80 amino acids, then typically there are too many proton signals to really resolve the spectra. And so people started to realize that if they wanted to go routinely do protein structures of proteins, say over 100 amino acids to keep it simple, then they had to introduce some other parameters in this. And so what they decided, since nitrogens and carbons are of course very important in proteins, they had to start isotope labeling the N15 and the carbon-13s in the protein as well. Now, in order to do that, you have to do bacterial expression of your protein. And so that's what we're going to talk about in the next 20 minutes or so, is how can you do bacterial expression. And if you're smart during bacterial expression of the protein, you can also put an affinity tag on the protein and purify it easily after you've made it in E. coli. Once you've prepared, and now we can do multinuclear NMR. Multinuclear means 1H, carbon-13, 15N, right? 3 different nuclei. Proton-proton would be homonuclear NMR because they are two of the same nuclei that we've been studying. Now, with proton-proton NMR, we can usually go up from 1D to 2D NMR, but with carbon-13 and 15N labeling, we can routinely do 3D and 4D NMR. So the spectra become more complicated because it's not just in 2 dimensions, but in 3 dimensions, in some cases 4 and even 5 dimensions. And then what we have to do is we have to make a backbone assignment. We have to assign what signal comes from what proton or what carbon or what nitrogen in the protein. And once we've got the backbone assigned, we actually, from the information that we get from this process, we already know what secondary structure is along the protein. We talked about that in the previous lecture. Amino acid side chains then need to be assigned. So we need to figure out for every amino acid side chain where its signals are. And then we can start with these structural restraints. And the most important ones are the proton-proton NOEs and those distances we have just talked about. And then we do the structure calculation and the refinement, as we have just discussed. And then the fun starts, because once it's done, you can start to look at all the interactions of a protein. You can look at the changes in pH of a protein. You can see what the protein binds. Does it bind to any other proteins? So then the real biochemical questions can start to be asked once you have the structure solved. Now this is shows you some of the steps. This is the NMR step. So this is the NMR sample. Usually, as I said before, 500 to 600 microliter, so smaller than a milliliter. This is a very peculiar moonlander shaped magnet, but this is a magnet that the manufacturer of this magnet no longer in existence, but some magnets actually last for 30 years, so they can be used for a long time. And so you can see, you can walk up the ladder and deposit your sample right up top in there. So teeny tiny sample here going into this whole super big magnet. And then you collect these spectra. So this is an example of a 2D spectrum, how it really looks, but then we look at it from the top. And so this is sort of the diagrams that you're going to see is one axis of chemical shift here and another axis of chemical shift here. Once we have labels on all these peaks to say, oh, this peak is that particular proton in the protein, we can start to measure these distances between all these different hydrogens. Those are these NOEs. And once we have all these NOEs, we can use those to as distance restraints to calculate the NMR structure. So this is the typical NMR structure as an overlay of the 20 lowest energy structures. So how can we now get these isotopes into the protein, right? Because remember, carbon-13 is normally only 1.1% naturally abundant. Nitrogen-15 is normally only 3.5% naturally abundant. And so if we want to bring that to close to 99%, we have to make a trick. We have to do something. So what we do, and this won't surprise you coming from the biochemistry program, is that we actually make the protein in E. coli. E. coli is a very good expression vector. And what we do is, here is an expression plasmid. So these are plasmids, so circular pieces of DNA, which have an antibiotic resistance gene, which allows you to select for them, right? Because only strains that have this plasmid, only bacteria that have this plasmid with this resistance gene will be able to grow on antibiotics, so other bacteria can't contaminate it. You need a promoter to drive the target gene expression, and then you put your target gene in there. So let's say this is your expression vector. Now, if you're smart, you say, I get my protein, but once I make it, how am I going to purify it? If I already stick a tag on this end or on this end, I can actually fish it out later much easier, right? It just becomes part of the protein. It's a fusion protein, right? And the most popular fusion is what we call the histag, right? You just put 6 extra histidines on the protein, and those 6 histidines can be recognized by a nickel column, and then you can fish it out. All right, so here is your plasmid, right? And then this plasmid, normally what you do is you have your plasmid here. There's something in here that you don't want to express, so you cut that out with restriction enzymes. And then you take what it's labeled here, PCR product. So this is something that you have cloned. These days, we actually just order synthetic genes. So we actually give an amino acid sequence, have the codons optimized for expression in E. coli, and 3 weeks later in the mail, we get a complete synthetic gene for the protein that we want to express. And then we ligate that into this expression plasmid. And then we put the expression plasmid into E. coli. And then we start to grow these E. coli on this medium, which has this antibiotic in it. So only the strains that have a plasmid with a gene inserted here and the antibiotic resistance will start to grow under these conditions. Then what you do is you pick up these cultures, these little spots of protein, and you send it for DNA sequencing of the plasmid. So you purify the plasmid, you send it for DNA sequencing, and then you hope that you get exactly the right sequence that you intended to make. So that's how you confirm that you got your target gene inserted in there. And once you've done that, you first do an expression trial. So you actually grow E. coli and you see if the protein that you want is actually expressed. And if you know that this is on a small scale, then you go to a big scale and you actually see that sometimes you can get a really large amount of protein produced. These plasmids are very efficient, so you can make a large percentage of the protein in E. coli, the particular protein that you want to make. One other trick that we all use is that we make the gene inducible, so it's not expressed all the time. So you use, what you do is you insert your plasmid into E. coli, then you grow your cells up to a certain density, and then you say, okay, now I want to stop growing because I now got enough cells to make my protein, and now we add the inducer. So the usual inducer is IPTG, which is a non-hydrolyzable analog of lactose. So there's a lactose regulator in the system, and you bind IPTG to it, and only then after the IPG sits on there will that gene get expressed. So you can basically switch on the production of the gene once your cells are at a high enough density. So this is now the most routine way in which most proteins for structural biology are produced. Now, okay, so I already told you if you were smart, you had made sure that there was a his tag either on the N terminus of the protein or on the C terminus of the protein. So now you can try to purify your protein, because your protein, here is a column that recognizes histidine. This is called a nickel NTA column. So here you see this is the nickel, which sits in some chelator, but only part of the chelation sphere of this nickel is fulfilled, and it likes to bind to the nitrogens and histidines. So if a protein with exposed histidines come by, it will interact with the nickel. And this is column material. So you make E. coli, you crack open E. coli, and all the proteins of E. coli are in there, and you pour it in this column. Now, essentially, everything is going to run through, except for the things that have exposed histidines, right? And so the his tag will bind your protein of interest to the column. And so all these other proteins, they all come through and they're washed away. And now your protein is stuck on the column. You see that here. Protein is stuck on the column. We got to get it off. So what we got to do is we got to bring in something that looks like histidine. And it's actually cheaper to use just imidazole than to use histidine. So you use a really highly concentrated imidazole buffer to knock your his tag, and which has the protein attached to it, off the column material. So here you use imidazole as a ligand, and as it washes through, you see it also washes your red protein off. And now your protein is usually already 95% pure, right? Many cases you don't have to do any more purification steps. This is often a one-step purification. All right, so his tags are extremely popular for purifying proteins. 80% of all the proteins that are deposited in the PDB as structures have actually been purified somewhere in their life as a his tag protein before they started doing the structural analysis. So here's another example illustrating exactly the same thing again. So this is a protein that expresses in humans and in other mammals, right? So this is an IPTG-induced culture of PET. PET is one of these expression vectors, and then his calreticulin strain. So calreticulin is the protein that they want to purify. So they inserted the gene for calreticulin in a PET expression vector, which already has this 6-his tag in there. So as you produce the protein from here, you end up with this. This is calreticulin. And here you have 1, 2, 3, 4, 5, 6 histidines sitting, in this case, looks like to be on the N-terminal side. But in other cases, you can also have the his tag on the C-terminal side. You can try both. Sometimes one works better, sometimes the other works better. So here you see your protein with the 6 histidines on it. You put it on that nickel column. Everything else runs right through because it doesn't have any affinity for that nickel column. And now your protein is still stuck on there. And now you come in with your high concentrated imidazole buffer. And what that does is it washes your specific protein off the column. And now in one step, you may have it almost completely pure. It doesn't always work, of course, in one step. Sometimes you have to do a second step. But in many cases, you can get well over 90 to 95% purity of your protein. And in many cases, that's enough. So this works for lots of proteins, right? And, and nowadays, people use synthetic genes because it's easier, because you can have the codons directly optimized in the synthetic gene for expression in E. coli. E. coli has slightly different codon uses than humans. And so if you just take a human gene and insert it, then sometimes the expression is not so efficient because E. coli has difficulty recognizing the codon. So if you can do a synthetic gene, it makes your life a lot easier. You don't even have to clone it. You just sit in front of your computer screen, you go to the protein database, you isolate the amino acid sequence of what you want to express, and you give that to the company. And as I said, 3 weeks later in the mail, for a couple $100, you get your synthetic gene. And you're off to the races with your expression. So this his tag strategy is really very, very important these days for making proteins. Now, we know now how to make proteins, but how do we put the label in there? Because we haven't talked about that yet. Well, if we want to do that, we have to put 15N in the medium, right? So that E. coli just takes it up. E. coli can synthesize all the amino acids. So all it really needs is a nitrogen source, is ammonium chloride, right? So if we give it N15 labeled ammonium chloride, all the nitrogens in the protein will become N15. Very easy. Now, normally, of course, we like to grow things in rich media because E. coli just likes to grow faster because it doesn't have to synthesize amino acids. It has all the goodies that it wants, so it can just take it up and grow fast. But if we do that, the only NMR sensitive nuclei that we're going to get are the hydrogens, because the hydrogens are always there. They come from the water and from the hydrogens and the soup and the glucose that the E. coli consumes. So if we want to have more than just the hydrogens in there, and we want to label, for example, all the nitrogens, we go to what we call minimal medium. Minimal medium is essentially just some salts, glucose, and ammonium chloride. That's really all you need to get the protein made. And E. coli grows a little slower, and maybe the expression levels are a bit less, but it usually does a very good job. And in fact, we have had a few cases where E. coli at minimal medium actually expressed better than on rich medium for reasons we don't know, and nobody's ever been able to explain to us either. But it's sometimes just one of these things that you find by accident. Now, if you want to also label the carbons, right? So you want to put carbon 13 here everywhere. See, carbon 12 is always there, but carbon 12 is not an NMR nucleus, remember? It doesn't have a spin, so you can't do anything with it. So you want carbon 13 in there, you know, replace the normal glucose that E. coli grows on and put carbon 13 labeled glucose on it. So it's totally carbon 13 labeled glucose, and it will get expressed now with all the carbons labeled with carbon 13 as well. Now, for bigger proteins, you actually have to remove some of the hydrogens. And this is done by growing E. coli in heavy water. Now you're asking a lot of E. coli. Not only is it in minimal media with N15 and with carbon-13, but now it's growing in heavy water, and that slows down E. coli quite a bit. So you usually have to train E. coli a bit by growing a few rounds in heavy water before it will do that. And it may not always produce exactly the amount of protein that you want. So you may have to scale up your production system a bit. But this is how you can label just about everything. And if you want to, you can also specifically label amino acids, right? You can just, for example, we like to work a lot with methionine. If we want to make methionine labeled protein, we just use minimal medium, and then we put methionine in the medium. E. coli knows it's there, so it doesn't synthesize it anymore. It takes it up from the medium, and then we can have the methionine labeled, and then nothing else is labeled. That's another way in which you can use specific labeling to do NMR experiments. So you have lots of ways to play with this. All right, so the most important experiment after you've done N15 labeling, and almost all papers that discuss structures and NMR interactions, they talk about this so-called HSQC spectrum. So HSQC is a big word which stands for heteronuclear single quantum coherence. So that's just a description of what really is being detected. And that's not so important to remember that. What is important is that you have to remember that it correlates the nitrogen and the hydrogen in the NMR spectrum. Now, where are that nitrogen and that hydrogen? Here they are. Here's an amino acid, right? Here's the alpha carbon, the alpha proton. This is the carbonyl, and this is the NH. This is the nitrogen, and this is the hydrogen. They label it HN because it's the hydrogen that's attached to the nitrogen. Now, if this is N15 and this is 1H, then they're both spin 1/2 nuclei. So think about it, a spin 1/2 nucleus is a tiny compass needle. Two magnetic things close to each other communicate with each other. So these two are coupled to each other. And so you can actually do a spectrum in which you have nitrogen on one axis and hydrogen on the other axis. So you correlate the nitrogen and the hydrogen spectra. And this is what we call the amide in the peptide backbone, right? There would be another NH here. If you continue with the peptide, there would be another CO here. The peptide can go in that direction. So you have many NHs in the protein, right? All right, so this is a picture of what here, you see that again. Here's one amino acid, here's another amino acid, here's NH, here's the previous NH, right? And there's a website that has a lot of information about this sort of stuff on there, so you can read up on there if you want to. But this is a typical HSQC spectrum, as you'll see in the papers. And this is the nitrogen axis. So this is the nitrogen shift that's on here. And on the bottom here, this is the hydrogen axis. So this is the amide hydrogen that's along here. And if you remember, that one appears somewhere between 6 and 10 ppm usually in the spectrum, if you looked at the notes from the previous lecture. So every cross peak in here represents one of these, right? Every combination of nitrogen and hydrogen may have a unique position. If you look along this particular proton chemical shift, for example, you see that as I go up with my cursor here, I hit several peaks. So in the proton dimension, all those peaks overlap. If I go in the nitrogen dimension, say along here, you see that there are a number of peaks that also fall and have the same nitrogen shift. But by defining the cross peak between the nitrogen and the hydrogen, you now get an almost unique position for every individual peptide NH bond in the spectrum. Now there's a couple other NHs in the spectrum. This one here is actually an NH in the side chain of tryptophan. They're usually here above 10, so they're easily recognized. There's one nitrogen in the ring of tryptophan that has a hydrogen attached. So there's 2 tryptophans in this particular protein because there's 2 of those peaks. And then these guys up here, they're actually not backbone amides, they're side chain amides. And then here, down here, you can sometimes see the arginine side chain, which is also detectable in these sorts of spectra. But most people don't look at the arginine side chain because there's not much structural information in there. So here's another HSQC spectrum. This is more what it really looks like in a paper. So again, if you look, this is the nitrogen chemical shift. So somewhere around 120 ppm on the nitrogen axis. That's where all this stuff is centered. And this is the amide protons between 6, 7, and 10 ppm. So this is the proton axis. This is the nitrogen axis. Now, if you look at this region here, you actually see that some of these peaks appear double. You can see they draw all these lines here. Now, why is that? Because this is actually the side chain of glutamine and asparagine. If you look at the side chain of glutamine and asparagine, there's 2 hydrogens on that nitrogen. So those 2 nitrogens, they're both coupled to the same nitrogen. So they should have the same nitrogen axis, but they have different proton shifts. So that's why you can draw a line through them, which is their nitrogen shift, but they have different proton shifts. But the backbones, so you can recognize those, and they don't really add much to your structure calculations. The one that you really want, and that's interesting, is this NH for the backbone, NH in the protein. And of course, if you have a protein of about 100 amino acids, you usually see about 95, 96 peaks. Why? If you have 100 amino acids, there should be 99 amides because the N-terminal amino acid is not an amide. The other thing is that prolines don't have an NH. Proline residues don't have an amide, and so they're not detectable. Now, the other thing is, if you have lots of NH, so big protein, you're still going to get overlap in this region. So this 2D strategy works well up to about 200, 300 amino acids. Now, another thing that's very useful about this HSQC spectrum is that it actually tells you whether the protein that you've produced is folded or not. So it gives you a fingerprint of the protein folding. And in that sense, it's a very useful experiment. And even protein crystallographers, when they make their protein recombinantly, they often go to see their NMR guy and say, hey, can you tell me if my protein is folded? Because some proteins, maybe if you made a mistake in making your construct, they won't fold properly. Now, this is what you typically get for a protein that is folded. It's well distributed. The peaks are all throughout this window along the nitrogen 15 axis and throughout this axis. But this is a typical example of a protein that's not folded. So everything is clustered in one region. So it's the folding, the three-dimensional folding of a protein that really gives rise to the giving different environments for every nitrogen and hydrogen that are involved in peptide bonds, and therefore they have different chemical shifts in this spectrum. So this gives you a good fingerprint to look at the folding of a particular protein. Right, and again, what we're looking at is we're looking at a labeled N15, and this is always a 1H, so this is the only bond that we're really looking at in here. Here you see some of these double patterns that we just talked about, which arise from the side chains of asparagine and glutamine. All right, so let me just close out then with a little example. This is an experiment we did a few months ago. We were interested in this particular protein called staphylokinase. It's a protein produced by a dangerous bacterium, Staphylococcus aureus. Nobody knows why Staphylococcus aureus makes this protein, but it was discovered many years ago that this protein, which is quite small, only 15.5 kilodalton, can be used for dissolving blood clots. And so people are quite interested in the potential of this. And what we did is we, of course, made this protein in E. coli, labeled it with N15, and it gives very nice NMR spectra. And that is in part because 15 kilodalton is a nice size for NMR work, right? It's just a relatively small protein. And what we were interested in at first is that there are actually two forms of a protein, where the first one, where the protein normally, when it's found, it starts at the N-terminus and then goes through this beta sheet, and then there's a helix, and then it ends up here at the C-terminus. These green and these pink things, they're important for interactions with plasmin and fibrin, which are proteins involved in blood clotting. Now, what was described in the literature was that if you cut off the 10 amino acids here, so this is the N-terminal 10 amino acids, protein becomes more active. And so this was the structure that was determined already in 1998 by a group of German researchers. It was an NMR structure, and this is what it looks like. And what we wanted to know is if you take off these 10 amino acids, is there a big change in the protein structure, or is it going to be exactly the same? Right? Can we take it off, and what does it do to the protein structure? So what we did was we did the following, right? This is normally SAC is secreted by Staph aureus, and it says that this is from this other group that said that N-terminal proteolysis would give it a higher activity. Now, normally, because it's a secreted protein, it has a leader sequence, of course, which during this processing gets cleaved off. So we're not interested in that. So when we want to make this, we make it in the cytoplasm of E. coli. That's where we have it expressed. So we made what we call SAC, which is the full protein from residue 1 to 136. And then we made another construct which started at residue 11, and then all the way till the end, SAC D10. It misses the first 10 amino acids. And so the question then is there a change in the protein structure after we remove these 10 N-terminal amino acids? And so, of course, we did this by NMR. So here you see our purification. We overexpress. We use a His tag to fish it out. You see the protein is almost completely pure. And this is the SAC D10. It's almost completely pure after you do this. And the important part is in this particular spectrum, because if you look at it, it says there are two spectra here, a black one and a red one. Now, if you look closely, there's a few black peaks that are extra. Which are those? Well, those are the black peaks for those 10 amino acids, the amides for those 10 amino acids, which are not present in this particular part, right? But if you look, all these red spots, almost all of them fall perfectly in all the black spots. So what does that mean? It means that there is absolutely no change in the structure of a protein after you take off the first 10 amino acids. So the protein completely maintains its overall structure after the removal of those 10 amino acids. So this is an example of how you can use HSQC spectra to look at protein folding, protein unfolding, and what happens to protein when you remove certain things or when you introduce a mutation or anything like that. So there's lots of examples like that in the literature. Okay, I think this is a good point for today to finish. And what we're going to talk about on Friday is that, of course, bigger proteins require bigger toys to analyze. And we're going to talk about what else you need to do, apart from bigger toys, to get into bigger proteins. So if anybody has any questions, please hang on, and I'll be happy to try to answer them after the lecture. Otherwise, just send me an email. I'm not always fast in answering emails. I still type with 2 fingers, but I do get by. Eventually, I might get to you. Okay, thanks, guys. See you on Friday.